Hello everyone, welcome back to another episode with the Life Science Doc. This week we will be covering science versus pseudoscience as well as the scientific method. So let's begin.
Here we go. As our title screen says, science versus pseudoscience and the scientific method. My goal is that this information helps you on your journey to understanding how to tell the difference between legit science and some shady things that people try to pass off as science. And in this process, help you to discover some of what this wonderful world has to offer. So first, a brief recap. In our first lesson, we defined science as the systematic study of the natural world. I mentioned it can be thought of as a tool to study and learn about the world around us. With that in mind, it's very important to look out for its sinister counterpart, the shady methodology known as pseudoscience. So, pseudoscience is a belief or practice that is based on incorrectly applied scientific methods. Because of its close resemblance, it can easily be mistaken for real science. However, we can tell the difference between them by looking at how experiments were conducted and how logic was applied in reaching a conclusion. Typically, with pseudoscience, the claims that are made cannot be tested and therefore cannot be verified. And if something cannot be tested, how could we ever analyze it for ourselves to see if it's true? So think of it this way. With pseudoscience, what you have is an instance where someone leapt to a conclusion without any empirical evidence and just holds tightly to that belief regardless of whatever else is out there. Remember our previous lesson on traits of a good scientist. You should always be skeptical and test things out for yourself to see whether or not they make sense. Here is a table that covers the main differences between the two. Notice that with science, we are willing to change our opinion of things when we see new evidence. Science undergoes peer review. We tend to take in account of new discoveries. We are open to criticism, so if someone speaks against something that basically that we're promoting, we invite it and we kind of break it down and see whether or not they're right. We have verifiable results. We limit claims of usefulness. Usually when we have studies, those studies tend to say specifically what things apply to, and we take accurate measurements. With pseudoscience, you'll notice that they don't like to change their ideas. There's no peer review process that goes on at all. Selects only favorable discoveries, meaning that if they have evidence that speaks against what they believe, they tend to discard it, pretend it doesn't exist. They see any criticism from people as conspiracy against them. Their results typically are not repeatable through experimentation. They claim widespread usefulness, meaning if they find something that does work, they say it works for everything. And they usually give ballpark measurements instead of accurate measurements. So with that, hopefully you have a better idea on how to spot the differences between the two. Now that we've gotten the science and pseudoscience stuff out of the way, let's move on to the methodology scientists use to learn about the world around us. As I have written here, knowledge is acquired through a process of investigation. We can break an investigation down into two parts: observation and experimentation. Observation is the process by which we obtain information using our five senses. I mentioned this once before using a household pet as an example. You can use all of your senses to describe the animal in great detail, which would then help others who have never seen it know more about it. And that way we can also make use of models. Models are representations of things that are either too large or too small or too far away for us to study properly with our senses. While not perfect, they still help us to understand the things we wish to learn about. The images on the left are of a cell model and a solar system model. Things are both too big and too small for us to observe directly. Can you think of anything else that you might use in daily life that is a model? Now we move on to experiments. Experiments are organized procedures to study things under controlled conditions. Now, we say controlled conditions because the outside world has many things going on in it. So typically, if we want to learn about cause and effect, we would need to isolate certain things to make sure that we can define what is influencing an outcome. Recall that in a previous video, I said that there are two places to gather evidence, in a lab or in the field. So experiments can be done in both places. And let's be honest, they can also be a lot of fun. Now that we've covered what observation and experimentation are, let's move on to the elements of an investigation. As seen here, we have three primary elements involved with investigating things. We have hypothesis, independent and dependent variables, and observation and data. And we will be discussing what each is about separately and as a part of the scientific method. So the first element is hypothesis. A hypothesis is a testable idea or explanation that we use to explain an occurrence. We can think of it as an educated guess. Let's take a second to understand that a hypothesis is not a theory. A theory in science is something that has been studied at great length with a large pool of data and evidence backing it up as an explanation. So when people say, I have a theory in everyday casual conversation, what they really mean is that they have an untested hypothesis. Now, our next element of investigations involves variables. We have independent and dependent variables. As you can see here, an independent variable is a factor that is being manipulated or changed on purpose in an experiment. And then the dependent variable is the outcome. It's the thing that depends on the independent variable. So as a great example, we know that plants and trees will grow based on the amount of sunlight that they receive. In this scenario, the amount of growth that happens depends on the amount of sunlight. So growth is the dependent variable because it depends on sunlight. And so sunlight is the independent variable because it's what we can control. The last of our three elements is observations and data. On the left is an example of data collected and organized into a table for easy reading. In my previous example, I mentioned sunlight could be our independent variable when discussing growing of plants and trees. In this situation, however, we're using sunlight as a controlled variable, meaning all plants in this experiment will get the same amount of sunlight. Instead, what we want to see is the effect of water on plant growth. So that becomes our independent variable. Growth still remains the dependent variable because that is the outcome. So I want you to understand this concept as it will help greatly when working on science projects in the future. Now, let's get into the actual meaning of observation and data. To observe something means to notice it and for it to have some sort of significance to you. Think of things that you are interested in, like cars, nature, or even a crush that you might have. When you look at them, you don't just see them, you take notice of them because they're important to you. Our next word is data. Data is the information that you gather through observation and experimentation. All those numbers on the left side of the table are our data because these were things that we could measure. It's important to remember that all data gathered in an investigation must be documented. This is what we use to draw our conclusions and serve as our evidence. If you discard or hide data, you become untrustworthy and come into the realm of being a pseudoscientist. Now that we have covered the basic elements, we can discuss the method by which all scientists figure out how things work. That method is called the scientific method. Scientific method is the process of objectively establishing facts through testing and experimentation. The basic process involves making an observation, forming a hypothesis, making a prediction, conducting an experiment, and then finally analyzing the results. So we will go step by step in how to follow all of these. In science, we may not approach each problem the same way, but it's best to keep in mind that the method I'm going over is a general guideline that can be used in most cases. Typically, after observing something in the world that we don't readily understand, our first instinct is to ask a question. This is how everything starts. We must first define a problem or state our inquiry. For example, let's say that we walk into a room, we flip the light switch and it doesn't turn on. Now we have a problem. The problem is we have no light. So the first thing that comes to our mind is, why isn't the light turning on? Now, after stating our inquiry, which was asking the question, why isn't the light turning on, it's time for us to form a hypothesis. Remember that a hypothesis is a proposed explanation, or basically an educated guess. These explanations must be able to be tested so that we can have some validity. Take note that a hypothesis will also be predictive in nature, so we assume that whatever test we perform will turn out a certain way. Going back to the darkroom example, we might say, I believe the power is out. This is a good hypothesis because it can be tested. It also gives us our outcome prediction, which would be that if the power is out, then the lights won't turn on in any room that we walk into. For the next step, we set up our controlled experiment. In the darkroom example I mentioned, we could go to any other room and see if the lights are on. If so, we've just proven our hypothesis false. If not, then perhaps we are correct and the power is in fact out. This example is fairly straightforward and simple, but many times in science things are not so easy. Remember, I mentioned we should have independent and dependent variables identified. So, independent variables include things such as equipment, temperature, time, or substances that are used, while dependent variables are always our outcomes, which is the things that we observe happening as a result. So, for this example, I've picked something a little bit more involved. This is how Edward Jenner figured out that if you expose someone to cowpox, that they would be immune to smallpox. Basically, he had heard that milkmaids who were around cows constantly and got cowpox wouldn't develop smallpox. So he decided to test that out. Since the hypothesis is basically that someone exposed to cowpox wouldn't get smallpox, he took pus from a pustule out of a milkmaid's arm and then injected it into a kid. The kid then got cowpox, and then afterwards he exposed him to smallpox and the kid didn't get smallpox. So that's how he was able to prove that getting infected with one disease made it so that you could be immune to a similar disease. So after we've performed our experiments, it's time to create a report of findings. All data that was obtained from the experiment should be collected and organized into charts, graphs, or tables so that we can analyze it and gain some meaning from what's going on. In this chart, the experiment measured the effects of different types of light on plant growth. Note how the different lights used have their own color to make it easy for reading. Now, our dependent variable or outcome, which is the plant height, is measured in centimeters and is plotted on the y-axis. Notice that time, which is clustered together with a type of light being tracked, is labeled along the x-axis, which is where the independent variables are plotted. From this chart, we could easily conclude that at 13 days of light exposure, grow lights and sunlight yield about the same result. However, if we look at the whole thing, we can say that grow lights help plants grow faster earlier on. This is why it's important to document everything, so that way you can see how trends go, and if you only have, let's say, 4 days to reach maximum height, you know which one to choose. So at this point we will be drawing a conclusion since we have all of our data organized into tables. I want you all to remember something important about this process. That is, being right isn't the goal. The goal is discovering truth. If our experiments ultimately demonstrate that our hypothesis was wrong, we simply state that fact and then we explain what we found to be true. After all, we're learning about the world around us, not declaring things that we wish to be true. Okay, so the conclusion was not the final step of this whole process. Pretty much once we get through the entire thing, it's time to repeat everything all over again. In our field, validity is the most important thing, and in order to demonstrate that, we have to make sure that our findings are reproducible. After all, it wouldn't be much help if something was only accomplished once and then it never worked again when you tried it. It's important to note that we don't just repeat things for the sake of proving that they work. Sometimes we'll make slight modifications just to kind of see how different circumstances yield different results. As I said before, the world is a big, uncontrollable place, so it might help to understand what limitations exist when we're designing the framework of our experiments. On the left, there was an experiment done to see whether or not that traits acquired during life could be passed on. This experiment was repeated over 22 generations, which gave pretty good evidence that cutting off tails of rodents um, does not produce kids that have short tails later on.
Well, that concludes this lesson on science versus pseudoscience and the scientific method. I hope you have a better understanding on how to spot things that are unscientific and how to set up your own investigations. Going forward, we will be diving into life science lessons. Some of that will include visualizations under a microscope and probably some experiments of growing organisms in petri dishes. So until next time, have a good one.